Okay, well, hello everyone. Thank you so much for having me here, and I'm going to talk to you about how to squash as many flies into the Darwin Tree of Life project as we can. So just to give you a quick background on the project. So the main aim is to sequence high quality reference genomes for all over 60,000 UK species. It's currently a network of 10 partner institutions, as you can see on the right. And the goal is to generate data that is relevant and of immediate use. So this kind of project has never been done before in the world. And so the reason British biota has been chosen in this pilot is because it's one of the best known in the world. They're really amazing, detailed location records, a thriving community of engaged naturalists, and the biota represents over a third of family diversity on Earth. So just to recap on the terms that I'm going to be using, genome versus DNA barcode. If we look at all these squiggly, colorful lines, these are chromosomes and they all represent a genome. And if we look at just one of those chromosomes, within that chromosome, we have genes, and normally a DNA barcode is a small section of one of these genes. So for a genome, as an example, for this Drosophila, it holds about 130 million bases of information, while if we look at a DNA barcode, it's about 600 bases. So it just shows you how much more information you can get out of a genome. And then to sort of show you the benefits of genomes and barcodes, with a genome, you generally do about one genome per species because it's quite a long, costly process, but it can give you improved predictions of the effects of climate change on biodiversity. You can use it with invasive species and get really detailed history and invasion routes for species, as well as looking into the adaptiveness of a species. Does it have the potential to be invasive if it's introduced somewhere? And then one of my absolute favorites is the conservation genomics. And with all this information for all these species, we can really understand the dynamics of genes that can help us avoid extinction, and then also high resolution species detection, tracking, and monitoring in the environment. And that includes abundance as well, which is really important for monitoring. And then if we look at DNA barcodes, you can have multiple barcodes per species. It's quick and easy to do, but it's also a very important first step in the genome sequencing process, because we use a barcode to sort of track that sequencing process through. The NHM is also currently building a DNA barcode reference library with verifiable morphological vouchers that will be curated into the collection. You can also map species distributions and populations with barcodes over wide geographic areas, which is very useful. And barcodes can be and are currently being used for tracking and monitoring and detecting species in the environment, but it's a lot less sensitive than using genomics. Okay, so back to the DTOL side and our goals by numbers. Our priority number one is maximum phylogenetic coverage. So in the phase one, which is where we are now, we're looking at families, and so that's representative of each family, and that would be 2,000 to 4,000 taxa that we would get genomes for. Phase two, we start looking at the genera, which is about 20,000 taxa, and then phase three, absolutely everything, about 60,000. And then other priorities that we include in all the phases, but in this phase as well, is research interest. If something is iconic, important, in whichever way, protected, endemic, and also, especially in the first phase, targetable. Is it actually feasible to find and catch, or is it thought to be extinct or very hard to find? And if we have a look at the Diptera, specifically, the scope for phase one is 150 species fully genome sequenced by 2022, but then we also need 850 waiting in the wings for phase two. So if we have a look at the global total of Diptera, there are about 188 families and 150,000 described species. We all know there's a lot more than that. And then in the British Isles, we have 111 families, which is a whopping 60% of the global total, which is amazing. And yeah, so that's the first phase. It's only 150, and then after 2022, it's going to go full steam ahead to do the other about 6,800 species in the UK. So if we have a look at, if we have a look at the total in process as of this month, we have 45 families in the bank, 154 genera, and 239 species. And we're also trying, well, we DNA barcoding every species that comes in, but we're also trying to get an extra species for a morphological voucher. So our barcoding total is 56 families, 200 genera, and 342 species. And so of our genome sequencing species, we have from White and Woods, let's go to the next slide. Yeah. From White and Woods, of those 239, we have 177 species collected from 33 families, including 55 species of hoverfly, which is quite amazing. If we have a look at the sample processing that we do, this is sort of a brief cap of what we look at, of what we do. So we have the specimen collected, we snap freeze it from live, is to be identified live at this stage. We dissect it cold, and so often with arthropods anyway, it's into head, thorax, and abdomen. We try and keep a morphological voucher as well. So if there's some diagnostic part, we take that. We DNA barcode the legs, and we need a very stringent tracking process because one specimen can technically be in 3 places at one time. And one of the really important parts of this is having these verifiable vouchers. So most of the time, because of this process and needing the whole specimen, we don't have an actual physical voucher. So sometimes this permanently stored, curated voucher is in the form of DNA. It can be tissue, it can be really good photographs, or in best case scenario, an actual diagnostic part of the organism. And then it's submitted for sequencing. So some of the big challenges that are going to be come up for Diptera are small species, smaller than about 5 millimeters, and species that can only be identified under a microscope as not alive. And so the solutions, we have a research and development group that are working on this, and another option which we can potentially use in the interim is actually using the high quality DNA barcode reference library to actually match the specimen to. So you wouldn't identify it, you just snap freeze it unidentified and then match it to the DNA barcode library later. So as we go on with the project, we're going to need some lists so people can see, the public can see what we've collected, how many specimens we've collected, and this needs to be readily available, especially in sort of the short collecting or fieldwork season. So we're working on these DTOL live lists. So the actual live list for DTOL is not quite ready yet, but here is a really good example from Ben Price at the museum called Fresh Base, which does include some freshwater Diptera. And so it's a Google spreadsheet that is linked to the data portal, the NHM data portal. And every time something goes into the NHM, it automatically updates this, well, every Monday. And the nice thing is that each one of these species names, you can click on it and it takes you to the portal. You can see where it was collected, who collected, and all the other information that's captured. So it'd be really great to have something like this for the whole of Diptera and the whole DTOL project so that everyone knows exactly what's happening all the time. So, and then speaking of timelines, which is a work in progress, we hope that things won't take as long as what I'm showing you here, but this is sort of what we're anticipating in the beginning while we work on making things quicker. So we want every specimen that is collected to be trackable, at least within the 2 months. But essentially, as soon as it comes into the museum and goes into our collection management system, it should pop up on those live lists. And then if your specimen is chosen to go to whole genome sequencing and is successful, this process can take up to a year, and you will be, so that's a year for it to be sequenced. Once it's assembled and annotated, then you will be an author on the published genome note. That can take over a year, depending. And then, of course, if your species was really small, it's probably going to be moved to phase 2, but that depends on the research and development that's done. If you collect a specimen and it goes for DNA barcoding, the morphological voucher will be curated into the NHM collections and associated with the project and the DNA. But that could also take up to a year, depending on how close to winter it is, because that's sort of when my team will have enough time to curate the specimens. But then the DNA extract from that specimen will go into our molecular collections facility, also trackable on the portal or on our live list, and that should be 2 to 4 months after we receive it. And then your barcode will also be uploaded to a publicly accessible database, and that can also take up to a year. So that sort of gives you an idea of how long at the moment, how long things are taking, but hopefully that time will be a lot shorter in the future. So DTOL 2021 for sampling, where possible, as I did mention before, we're trying to collect more than 1 specimen of each species, some for genome sequencing. Also, if it's very small, we may need a bit more tissue, but at least 1 for genome sequencing and at least 1 for DNA barcoding, so we can have this morphological voucher. We plan to run BioBlitz events across the United Kingdom, and the NHM are also receiving live specimens via post. So do contact us for tubes or other information. And we also want to do some smaller targeted field trips as we go, but it just depends on what we've collected, and we'll have to play that by ear. We've also put together an expertise survey, so please do fill it in if you can. It's in green at the bottom, tinyurl.com/invert-experts. And we would like to know your area of expertise, your taxonomic groups, and we'd like to keep you updated on field work and potentially our live species lists. And you can also suggest your favorite fly for sequencing. And then another invitation to the amazing Whiten Woods by Liam. It is an amazing place with a great research station. Looks like it's been refurbished quite recently as well. So it's one of the sites of the longest ecological studies in the world, and really a good spot for a potential field meeting in 2021. And if anyone wants to do any ad hoc collections, please contact Liam on the email. And then just to tempt you further, he's included some really nice pictures of some really nice flies. And with that, I would like to thank everybody who's involved directly with the DTOL project. A long list of people are working together to make this happen, which is quite amazing. And then, of course, a special thanks to those who've sent specimens in, especially during the pandemic and lockdown, and then our little field trip to Hart's Lock. So thank you so much, Mike, Ryan, Chris, Matt, Sam, Mark, and Duncan. And then there is the link to the survey in green as well, to remind you. Great, thank you, everyone.
Thanks very much. Thanks for that. That was lovely. We have some questions that have come in now, which is great. I'm just too busy thinking about what my favourite fly is, so you completely distracted me. Thanks for that. Okay, from Sue Taylor, we've been asked, how do you screen out pathogenic fungi and bacteria?
I think that would be a really good question for Mara, who's speaking next. Sorry, I don't think I can help you with that.
This may also be a Mara one then as well. Does it work for killed pin specimen as well for live specimens?
Well, for DNA barcoding, killed specimens and dry specimens is possible, but it isn't for you wouldn't be able to get the genome out of a dry specimen. It has to be live and snap frozen quickly as possible.
So you're getting lots of people to send specimens in the post?
Yes, yeah, sending them live. Sort of not that successful all the time. Depends on the weather, I think.
Yes, quite adverse at the moment. Do we have any more? Do you take photos of specimens before assembling? And that's from Alexander Butler.
Well, we take photos of the specimens before we dissect them, if that's what you mean. So we do take a sort of high resolution, try and do a couple of views, and hopefully catch the diagnostic part. But we're still working on the SOPs to figure out exactly which part of every species is diagnostic before we photograph it.
That's going to be quite complicated, I presume.
It is, yeah. We'll have the long version of the SOP and then the short.
Yes, yeah. How about international cooperation? Are there similar barcoding projects in Germany, Norway, etc.? And that's from Paul Burke.
For barcoding, yes, and that is something Mara will also mention in her talk with international collaboration. But I think for the genome work now, this is the pilot. So hopefully we figure out all the kinks, and then it will be easier for everyone else to pick up and fly with it.
Oh. Okay, so there's a lovely one here from I'm a student from, and they don't say, Demuni Hashani, but they would like to join in with your research work. So how does everyone help? I presume in the UK it's going out and finding new fresh flies and identifying them.
Yes. So we definitely rely very heavily on the community of experts, and that's going to be the success of this project, is having everybody working together. Anyone who wants to get involved and you're in the UK, you can email, maybe I should put an email there, but anyone from Darwin Tree of Life, there's contact at darwinTreeofLife.org. I think, is that right, Maura? Yes. Or any of us at NHM, Zoe as well. I mean, she can also, anyone really, contact anyone. We'll find the information that you need. Yeah.
And I love this one from Donald Smith. Why so many hoverflies? I think it's a question we all ask.
I don't know.
I think there's just a lot of them.
There's just, that's it.
Yes. Okay, let me go through. If we collect specimens other than the flies, are you interested in those at present? And that's from Bill Irwin.
Absolutely. If it's identified, we are definitely very interested. We're doing everything. The survey link is also to all invertebrates of the UK. So that's marine, terrestrial, freshwater. So it's not just flies. So.
But obviously the flies are the most important.
Of course.
There we go. That's what we want.
And I don't say that at every meeting.
Of course you don't. Nicely answered there. Do we have any more questions from the panel, or anyone else would like to make any input?
Erica, have you seen this last one? Does the post office question buzzing parcels?
No, I guess it's just come in. Brilliant.
Well, actually, it is. We have checked. It is legal to send live insects in the post. So they don't question it. They're technically okay with it. I think you have to write it on the outside, and then it's fine.
I think most people who've got cockroaches and things like that have sent many a live insect in the post.
Yeah, I think so.
Oh, there are a few more. Oh, it's gone. I will quickly just ask you, what's been the biggest challenge so far in gathering DNA-ready specimens? And that's Stephanie Skip.
I know you, Steph Skip.
Hi.
Sure. Biggest challenge of actually collecting. Wow. I actually think it was getting permission to put dry ice into the van so that we could take it into the field and process.